I just want to welcome everybody back to our hemp production webinar series today. We really appreciate you joining us. I know people join from all over the country. So in New England today, or the Northeast, it's unseasonably warm for the end of May, but I guess we'll take what we can. So I'm sure a lot of people are out in the field trying to get field work done. But those of you who were able to pull away today, we really appreciate it. And I think, as everybody knows, the webinars get archived and made available to our hemp listserv so that people can watch later when they have time. So thanks again for joining us. This is our third webinar this season, and we're going to continue adding webinars to the series. Alex Escher is going to be talking about building with hemp in late June, and we'll be adding on some more webinars as the season progresses. But for probably the next month, we're going to take a little break so everybody can get their field work done. And then we'll jump back into webinars again as the season progresses. And who knows, we might be able to get something in person a little bit later this growing season. All right, well, today joining us is Seth Crawford from Oregon CBD, and really appreciate you joining us today. I'm sure you folks are busy as well, but you're just enjoying your breakfast, I guess, and morning coffee. So good way to kick off the day. And we're so excited to hear about the new seedless triploid hemp varieties that Oregon CBD has been working on, I'm sure, for quite a while to get out to market. We're excited about them here in Vermont, and we're going to be trialing some of those in our evaluation trials this year. So we'll be excited to share performance data for the Northeast this winter. So with that, Seth, I'm going to just turn it right over to you.
Thanks, Heather. Really happy to be here. Just also want to say thanks to everyone behind the scenes for having me here today, both at the University of Vermont and at Oregon CBD. It's fun to be back in Vermont, even if it's just virtual this time. I'm pretty sure Vermont is my favorite non-Oregon state. My first trip there was in 2018 to speak at the Vermont Hemp Fest. This is right as the industry was really ramping up and in the midst of the first really big harvest of our varieties outside of Oregon. It's an unbelievably beautiful state, especially when it stays warm through September. My wife and I just really enjoyed our time being there, despite just missing Bernie Sanders seemingly everywhere we went. The place and the event, the Vermont Hemp Fest itself, were memorable to say the least. Someone messed up and let this particular cannabis-related event take over the swank Burke Mountain Resort. Beautiful location. But, if you've been to a hemp conference before, you kind of know to expect a certain carnival-like atmosphere. There's lots of vendors, the usual mix of pertinent and extraneous information, usually in unequal doses, tons of cannabis products, and a bunch of really interesting people. I met the two most memorable for me from that trip back to back. It was Tyler Heider and Sarah Schlosser. Both great people, but farming hemp in very different ways. Tyler helped start Northeast Hemp Commodities. It's a large farming operation based in Middlebury. They were just about to harvest their first really big crop, and he was sweating bullets about drying capacity. We talked about plant finishing times, fans, flow rates, temperatures, what you could reasonably expect to accomplish, and when things inevitably get unreasonable on the farm, just how many hours really are in each day. He needed a way to stagger his harvests throughout summer and fall to maximize efficiency before those inevitable frosts set in. So we've worked with him and his company over the years to help match the right flowering time and dominant cannabinoid to their at-scale farm approach. And in that process, learned just how unique microclimate effects can be in Vermont and how volatile the larger cannabinoid market in general can be. Sarah Schlosser, a true dirt under the fingernails, salt of the earth organic farmer. She patiently waited through the crowd to find my wife and I after the keynote, right after I'd caught my breath from strategizing with Tyler. I say this because we could see her working her way through the pack of people towards us because she had a freshly cut, 3 foot tall, fully flowered cannabis top sticking out of her backpack. I thought that sort of thing only happened in Oregon, but that's obviously not the case. So well done, Vermont. Well, Sarah literally had the very first lifter plant harvested in Vermont in that backpack. And she just wanted to say thank you for making a seed that finishes so early and so well in her particular location. Her and her husband Bob run Sandywood Farm. It's a diversified and constantly evolving small operation in Walcott. You can visit their website to buy maple syrup, diverse hemp flowers and products, sign up for a CSA subscription, or even book your wedding on their beautiful farm. They have my youngest stepson hooked on Sandywood Farm's pure Vermont maple syrup for life. Small farms like Sarah and Bob's need constantly evolving flower flavors, novel cannabinoids, and predictably early finishing times, as well as bulletproof performance to be able to ensure diverse offerings to their customers. Tyler and Sarah are two very real, very cool people, but they really also personify divergent approaches to hemp farming. Our breeding work is inspired by farmer stories like these, finding the overlaps and beneficial traits and identifying unique needs within the industry helps drive our research and development process. These areas of need are most often brought to our attention by the farmers themselves. We're tremendously thankful to the hemp community for their support over the past 6 years and really excited to continue pushing the boundaries of what's possible with this plant. Heather gave a talk at that same Vermont Hemp Fest as well, and it was literally the most packed and popular seminar of the entire event. The comments from people leaving that room were inspiring. Everyone loved it because for one of the first times in our collective adult lives, a real scientist was in a room talking to the public about cannabis production. Not don't do it, but this is what you need to consider. Here's what works and what doesn't. Here's what we know and what we don't know. By listening to farmers and applying best scientific practices to this particular crop, I think we are all collectively reinventing how cannabis is farmed in a very short period of time. We're grateful to Heather and other UVM extension staff for helping to push cannabis science and agronomy forward. To us, one of the best and most useful summaries of the past 6 years can be found on the UVM Extension Service website. There are 8 new 2020 research reports available detailing nutrient needs, flowering times, pest and pathogen resistance, and overall variety productivity evaluations for fiber, grain, and cannabinoid-rich hemp cultivars. This is the foundational work that allows farmers to make good decisions regarding best practices for their specific area of the state and to identify varieties that will maximize production efficiency and minimize the impact on the land. Tremendous thanks to Heather and her team who made this evaluative work available to the public. The reports, we believe, are mandatory reading for farmers, and we really want to encourage everyone to head to the UVM Extension website to check them out firsthand. Well, it's probably no surprise to a lot of farmers in Vermont that our varieties did well in these trials, but we were really excited to see Lifter rise to the top based on its performance. For overall flower yield, Lifter averaged nearly 4,000 pounds per acre, which is a phenomenal outcome considering that it finishes very early in the fall relative to non-Oregon CBD varieties due to our hybridization process. Only Suver Haze outperformed Lifter in average total cannabinoid content for this study, while all of our varieties delivered great results relative to the others evaluated. Suver Haze averaged 15%, and Lifter clocked in at 14.1%. Our white CBG was also the top performer in the type 4 pure CBG category. So I went ahead and did some extrapolations based on the data presented in these reports to produce the following graph, which shows total CBD yield per acre for all the varieties included in this study. This is a really good piece of data for farmers who are growing specifically for biomass, where per acre productivity is really the most important, it's the primary variable determining success. There's really no surprise here, but Lifter also wins by a substantial margin in this category, with over 555 pounds of pure CBD per acre, and Suver Haze is second at 431 pounds per acre. I think it's worth mentioning that this incredible biomass producer, Lifter, is the very same variety that won the 2021 High Times Hemp Cup last week, and multiple other awards over the years for smokable flower, which is, to our admittedly biased position, something that demonstrates that it's the high watermark for both biomass production and trim flower to emerge out of the first era of what we call the U.S. hemp reintroduction. While we're ecstatic about sharing these results with the world, it's really important to note that our first commercial trial of Lifter happened in 2017, and we've come a long way since then. We're excited about the next bevy of advancements and how they will help farmers attain success in an ever-changing market. And so today's talk is really providing an overview of what we believe is going to be the next step in the evolutionary advancement of cannabis with polyploid cannabis varieties. Lifter, even though it has done so well in these trials and really helped farmers achieve great results, we're looking at a diploid production version. That was what was tested. And we're really excited to see what the test results come back from multiple universities over this summer, what sort of weights and yields they end up getting with the new triploid improved varieties. So today's talk will provide some background on our company, how we develop commercial varieties, the processes we use to accelerate developmental timelines, and how we're applying modern polyploid plant breeding techniques to further improve our varieties. And I want to make sure that we have some time at the end for any questions that inevitably come up for anyone to make specific requests for traits of interest we should be working on, or just general questions about the polyploid work. So my brother and I co-founded Oregon CBD in 2015. We're Oregon's oldest continually operating hemp farm and the nation's leading supplier of feminized cannabis seed, with over 55,000 acres of our varieties grown in the U.S. since our inception. Farmers around the country have used our seeds to win over 40 different awards in trim flower competitions. Several of our cultivars, Lifter and White CBG in particular, are being used as the benchmark in university field trials when assessing new varieties. Our 49-acre research campus is located in the Willamette Valley and is home to 38 cannabis scientists, chemists, and horticultural professionals. We operate one of the most advanced cannabis breeding programs in the world at our little farm and collaborate with other scientists to better our collective understanding of this incredible plant. Today, our campus consists of over 275,000 square feet of sealed, climate-controlled next-generation greenhouses, over 600 isolated breeding chambers for targeted inbreeding, tissue culture facilities, a ploidy manipulation lab, full HPLC and GC testing, a high-throughput molecular lab, and a whole genome sequencing operation that has built the largest collection of high-quality whole genome sequence data for this particular organism in the world. We're light years away from the used shipping container and the handful of grow lights we started out with in 2015, and have both farmers and our talented team to thank for that growth and development over the years. Those rapid changes in growth highlight the fact that we're basically running on a treadmill to constantly improve our work using the best practices and technology available, trying to do about 10 years worth of work every 6 months or so just to maintain our competitiveness in an ever-shifting market and regulatory environment. Our goal has always been to produce consistent F1s for farmers. Our initial formula relied on a classic plant breeding strategy of pairing two highly inbred but unrelated parents. In our case, one a photoperiod sensitive plant and the other a day neutral variety, also commonly referred to as autoflowering. We do this to maximize heterosis or hybrid vigor. This approach requires us to separately inbreed the two parental lines repeatedly until lines are homozygous for our desired traits, and then combine the two lines to make F1 seed. We accomplish the inbreeding component by using our isolation chambers to self-pollinate hundreds of candidate plants at each stage of the process and evaluate using HPLC, our noses, and phenotype assessment. These are 4 foot tall, 2 feet wide, and 2 feet long, small grow tents outfitted with negative pressure HEPA filters and powered by small LED grow lights. We kill the undesirables and keep the winners, repeating the process multiple generations, generally until individuals become pollen sterile, and then trialing the final crossing combinations. With our traditional diploid breeding program, it takes about 3 years from the initial crosses for trait introgression until commercial release of new varieties. This approach ensures that farmers are getting a uniform and predictable crop that outproduces the competition, while also throwing a wrench in attempts by others to breed with our hybrids. F1s are very stable for farmers, but inherently unstable as breeding stock. This process takes a lot of time and a lot of space, though quite a bit less than what's required of many other traditional agricultural crops. Developing varieties with new, useful agronomic traits, predictable flowering initiation, different dominant cannabinoids, or novel olfactory presentations are all examples from our early work that continue today. This was initially completed through an expensive and time-consuming brute force approach that required large grow-outs, thousands of cannabinoid tests, and often fortuitous timing. Once enough chemotype and phenotype data was collected and new technologies became available, we began the process of identifying predictive genetic markers to rapidly screen large populations of seedlings. Today we have a number of key markers that help save us time and space by eliminating unnecessary plants shortly after seed germination, which allows us to evaluate ever larger population sizes at each stage of development. Marker discovery has been made possible through genome-wide association studies, cannabis-specific SNP chip panels from both Eurofins and Lighthouse Genomics, and our own targeted in-house whole genome sequencing. Many of these markers, while predictive, are not universal. Cannabis is a very polymorphic species, and our molecular lab has to engage in a lot of primer redesign whenever a new individual is introduced to the breeding program for trait introgression. Despite the inherent technical challenges, we're happy to report that the process has proven both manageable and groundbreaking, though it requires a lot of high-quality whole genome data wrangling. I say that as a person who is not responsible for primer redesign or our sequencing efforts, which would make this a great time to publicly thank our Chief Science Officer, Dr. Andrea Garfinkel, who drives our molecular lab to success, and to our genome lab director, Aaron Tripp, for making all that PacBio hi-fi whole genome data possible. Two really useful real-world examples of this are the discovery of autoflower markers and varin cannabinoid markers, which were identified using two separate discovery approaches, one from Inrgene and the other from Eurofins, applied to a unique population of plants. The two parents of the population could not be any more different for this project. One was a very tall type 1 varin-rich female that produces long spindly flowers with a very open structure. It doesn't initiate flowering until day length is reduced to about 11 hours, and then it takes about 16 weeks to fully mature. The other parent was a short, day-neutral type 3 plant that produces dense and aromatic flowers. Its life cycle from germination to harvest is 10 weeks total. We created a large F2 population by self-pollinating a selected F1 derived from the two parents. Suffice to say, the resulting F2 population genotype for that project contains a tremendous amount of diversity, making it a perfect candidate to discover important traits. The parental lines, F1 individual and 300 F2S1 individuals, were subjected to sequencing and analysis after we collected phenotype and chemotype data. The data was run through a multi-locus mixed model regression package in R to identify candidate markers, which yielded several high-quality associations for both traits. These are relatively rare traits. The autoflower response behaves as a single locus recessive trait, appearing 25% of the time in F2S1 populations when one of the original parents is an autoflower and the other is not. The varin-rich cannabinoid chemotype occurs 7.3% of the time in the F2S1 populations when one of the original parents is varin-rich and the other is not. Finding a varin-rich auto combining these two traits in the F2S1 generation, even with purposeful pairings to maximize trait introgression success, happens less than 2 times for every 100 plants grown. To identify 100 individuals with those same traits using classic brute force methods would require 5,480 plants to be grown out under 24 hours of light until the day-neutral plants begin flowering, followed by 1,370 HPLC tests to identify the 100 with the varin-rich cannabinoid fraction. It's about the equivalent of 46 days of continual chemical testing. Now we can identify those 100 individual standouts while they're still in 72 cell trays, only transplanting the winners for further inbreeding and evaluation, and not prematurely initiating the autoflower response through stress. It also helps keep our chemist, Matthew Otten, a little bit more sane. Even with these advancements, it still takes significant time and resources to fully introgress a new trait into our breeding germplasm before the resulting seed can be produced and released to farmers. The greatest benefit of marker-assisted breeding in cannabis so far is to increase the likelihood of finding outstanding plants. The day-to-day grind of the underlying breeding work hasn't changed, but we have added an extra step to our process with our Polyploidy work. So using this marker-assisted breeding approach allows us to maintain that same 3-year development timeline while adding additional complexity and features to the resulting product. So I'm hinting at polyploids here, so it's probably time for a quick interlude. We're going to play a little game called Guess the Polyploid.
Pictures of real life cannabis plants, and what I'd like viewers to do is write down either yes or no if you think the plant is a polyploid. For extra credit, when you see a polyploid, provide some rationale as to what visual cue tipped you off. You guys ready?
Yes.
Those 3 are on the screen, A, B, and C. What do you think? A polyploid or not? Is B a polyploid or not? Same thing for C.
So people are typing stuff into the chat box, Seth. I don't know. You probably can't see it when you're in full.
I can't, unless yeah.
Do you want people's answers, or do you want to just wait to the end?
Let's hold off just for a second.
Okay,
go ahead and,
sounds
go
good.
ahead and write them down, and then let's go back over them. So here's your next 3. You guys ready? Next 3 are D, E, and F. All right, we've got the next 3 coming up. E, H, and I. And the last 3, J, K, and L. Run back through these real quick. What does everybody think for A?
Looks like we have a couple of guesses for C in this slide from the chat box.
Okay, okay. But everybody's thinking A and B are not polyploids. C is a polyploid.
Yeah,
Okay.
everybody's saying no, no, yes.
Okay, so in this one, A is a diploid. This is not polyploid. B is also not a polyploid, and C is not a polyploid. So in A, this is all of these pictures were taken off the internet for specific mutations or a description calling these plants polyploid. And A is literally just a regular cannabis plant. B is an example of webbing, a webbing mutation happening in the leaf structure itself. And C is actually called fasciation. So this is not an example of polyploidy, but the most often confused example of what a polyploid is. So this is just a somatic mutation. In cannabis, we found that this is actually a heritable trait and can be passed on. It's fairly interesting. How about for D, E, and F?
All right, do people want to put in the chat their guesses for this slide?
No brave souls are guessing yet. Oh, we got one.
like Robert Shipman thinks that D is polyploid. I can see that now. That's awesome. And looks like Baxter, you're right. It's no, no, and no. That first one is another example of a fasciated plant. And if you look really closely, you can see the stem is really wide and very wide across and very narrow in depth. So this is another example of fasciation. It's just that the flowers on the top of the plant didn't completely fill in the same way that commonly fasciated plants do. So again, this is just a diploid plant with fasciation. E is a common mutation that we see in a variety called freak show. This is started off as a THC plant, but it has a really interesting mutation to the leaf that creates almost a fern-like appearance. And F is a great example of whorled phyllotaxy. This is multiple leaves emerging from the same growth point on the stem. How about G, H, and I? What do you guys think? H. H is that's just a flower growing on a leaf. It's abnormal, but it is not polyploid. So G is an example of what's called, this is an Australian variety, designated as ABC. It has a very interesting leaf mutation. The flowers end up forming normally, but the plants stay relatively small. So this is another example of a non-polyploid mutation that happens in cannabis. And I is commonly referred to as irrigation. This is basically a loss of the ability to produce chlorophyll in about half of that leaf. And again, not a polyploid. Now for the tough one, let's look at these last three, J, K, and L. I'll give you a hint and tell you that all three are polyploids, but different ploidy levels. So in this case, J is a triploid. This is actually our new sour lifter Seedless, triploid variety that we just released this year. K is a tetraploid, type 4 CBG variety. And L is a pentaploid. So this is one of our higher levels of ploidy that we're doing some experiments with. How many of you got them all right? It's a tough one. So one of the biggest misconceptions that we have in sort of cannabis writ large right now is that a lot of people mistake polyploids for fasciation. So the only way to really be sure about an individual's ploidy is to use flow cytometry to screen the plants. The term polyploid has unfortunately become a catch-all to describe any visual abnormality on plants found by a lot of home growers. So we're really trying to make sure that people know that those are just mutations that happen. It's not connected to ploidy. So that'll help us kind of get started here with the ploidy side. And I'll move through this quickly just because I know we're getting close on time. Let's contextualize the term seedless triploids. So these are, in short, cannabis plants that are incredibly resistant to pollination and contain other exciting attributes brought on by extra genome copies. Our development of triploids was born out of the trauma of losing several crops to cross-pollination events in 2016 and 2018, and hearing horror stories from growers year after year. If you've grown cannabis, you likely understand how devastating pollen drift can be on crops, and that it can travel hundreds of miles in the right conditions. Fully pollinated plants produce 50% less flower weight and 30% less cannabinoids. A seeded crop is essentially worthless if you're growing for trim flowers, and still devastating if your end goal is biomass or extraction. And as we've also seen in the first 6 years of production here in Oregon, feral populations of cannabis are regularly establishing themselves in fallow fields and have become a big enough concern that our state Department of Agriculture is now working on rules and penalties for landowners who fail to control rogue plants. Having a plant incapable of being pollinated fixes all of those issues and threats and lets farmers put their energy into growing great crops instead of worrying. To understand triploids, we need to establish some terminology. So chromosomes are packed together collections of DNA, which is the underlying genetic instructions for life. It's helpful to think of chromosomes as books of code, which make up the library that is an organism. Every species has a unique number of books in its library. Humans have 46, naked mole rats have 60, and cannabis has 20 in the diploid state. This library size is referred to as the chromosome set number. Ploidy refers to the number of homologous sets of chromosomes in an organism. So organisms with a single set are referred to as haploid, 2 sets are diploid, 3 sets triploid, 4 sets tetraploid, so on and so forth. Any organism with a ploidy higher than 2 is referred to as a polyploid. In sexually reproducing species, 1/2 of the chromosome sets are inherited from each respective parent. For humans, this means our individual genome is actually a fusion of 2 different genomes cut in half during reproduction. We're truly 1/2 of both parents. The same is true in diploid plants. While most vertebrates are diploid, many plants are polyploid. These include major agricultural crops of critical importance to humans, such as coffee, bananas, strawberries, red wheat, oats, citrus, potatoes, and tobacco, to just name a few. The main point to understand is that polyploidization is a natural phenomenon that's occurred many times in nature and has led to the creation of the ancestors of many modern crops. Polyploid induction in nature tends to correspond with extreme climate events or natural disasters and lead to increased diversity within a species. The only documented naturally occurring tetraploid cannabis population was found at 14,000 feet in the Himalayas by an Indian research effort about 10 years ago. The extreme climate and altitude may have been the causal agent responsible for that particular event. We can think of this as nature's way of creating options for diverse responses under changing circumstances. It's also important to note that these events are not changing the underlying DNA or introducing new genes. They are simply whole genome duplications of what was already present. In the case of polyploids, more gene copies tends to confer better adaptability and resilience. Plant breeders became aware of the remarkable superiority of polyploids in the early 20th century and have induced polyploidy and used natural polyploids in many ways to obtain increasingly improved plant cultivars. Some of the most important consequences of polyploidy for plant breeding are the increased size of plant organs, so bigger flowers, bigger leaves, bigger stems, the buffering of deleterious mutations, increased heterozygosity, and superior heterosis or hybrid vigor. Cultivars of non-cannabis plants have been generated with higher yield levels, improved product quality, and increased tolerance to both biotic and abiotic stresses. The moral of the story here is that polyploidy is a tried and true method of improvement in many crops. And an important bonus is that plants with odd chromosome set numbers, so triploids with 3, pentaploids, 5, and so on, often prove to be infertile. This is what makes seedless watermelons, bananas, hops, and now cannabis a reality. The first comprehensive chromosome scale cannabis genome was published in 2020. There's several other lower quality assemblies publicly available on NCBI, but the CS10 assembly at this point is considered the reference. It tells us that the cannabis genome size is approximately 800 megabases in size as a haploid, so that's 1 genome copy, and 1.6 gigabases as a diploid with 2 genome copies, and that's the natural state. The plants that we've all historically grown contain about 60,000 genes, with around 72% of the total contents in that genome existing as non-coding repeat elements. Cannabis's closest relative, hops, is about 3 times the size, with similar levels of non-coding repeats. Advances in genomic sequencing technology is nearly to the point of allowing us to create fully phased de novo diploid assemblies for the first time, which is going to be a tremendously useful tool for bioinformatics-assisted breeding programs. Our polyploid development program was designed and led initially by Dr. Sheng Chen, an OSU PhD graduate who's now an assistant professor at NC State. We still work with them and collaborate. Today, our lab is run by Brendan Rojas, another OSU grad. He's doing some really fantastic work as well. Our goal was to convert our diploid breeding stock into tetraploids and beyond test the fertility of various ploidies in controlled environments, and to test the agronomic traits of various ploidies via controlled crosses and initial field trials. Controlled genome doubling events require initial plants, a cell division inhibitor like colchicine or arizoin, steady hands, patience, and a flow cytometer, which is a really cool piece of equipment capable of measuring an organism's cell size. Hundreds of asexually propagated clones of a target plant genotype are pruned back until a single growth point remains. Then, in our case, colchicine is applied. Most plants die within a month, and the few survivors are screened by flow, and any confirmed tetraploids move on to the stabilization process, which takes about 8 months from the initial treatment. At each step in the process, plants are rescreened over and over as they have a tendency to revert to lower or mixed ploidies until fixed. If a plant has 8 shoots, all 8 shoots are tested. This continues until stability is attained, then the plants are ready for seed production, but not before a final flow screening. Tetraploid germplasm is always maintained in a separate greenhouse to avoid mix-ups, and we've now successfully created tetraploid versions of all of our classic CBD and CBG varieties, as well as several of our new CBDV production mothers and type 1 plants in our germplasm library, and have incorporated the streamlined conversion process that we've developed over the last 3 years into our breeding program as an essential step. This is cool. You can see in these images the resulting peaks generated by the flow cytometer when testing plant material of various ploidies, as well as microscope pictures of their actual chromosomes. So using multiple methods to validate ploidy levels is especially necessary when creating baselines for further analysis. But the chromosome pictures really help to convey the cellular changes present in polyploids. There's really a lot more genetic information in these plants, and you can see it when you get close enough. Diploid cannabis plants contain about 60,000 genes and have a genome size of 1.6 gigabases. Triploids contain 90,000 genes and have a genome size of 2.4 gigabases. Tetraploids contain 120,000 genes and have a genome size of 3.2 gigabases, or roughly half the size of the whole human genome. These extra gene copies are what researchers believe drives resilience and adaptability in polyploids. Seedlessness in triploid cannabis could really be a wonderful thing, but we had to design experiments that capture this essential quality in a meaningful way. To do so, we created 6 crossing combinations to assess seed production and viability. Diploid, triploid, and tetraploid versions of the same F1 hybrid cross were pollinated by diploid and tetraploid pollen donors. These trials were conducted in sealed greenhouses, and the target plants were subjected to massive amounts of pollen. This is intended to simulate a flower crop being grown side by side with a 100-acre grain farm. The results show that diploids pollinated by diploids produce about 2 times as many seeds as tetraploids pollinated by tetraploids, suggesting that ploidy increases result in lower fertility in cannabis. Tetraploids are not viable as a pollen donor for diploids, but are able to induce many problematically empty seeds. The key finding is that seed production is essentially non-existent in triploids when pollinated by either a diploid or a tetraploid. Even under substantial duress, they remain nearly seed-free, with a maximum average of 6 seeds per triploid plant when pollinated by diploids in a controlled pollen inundation zone. Triploid pollen donors are not reported in our upcoming publication because they are sterile and do not produce viable pollen, which has been confirmed in more recent experiments that we've conducted. In addition to relative seedlessness, triploids are also superior in many key agronomic traits relative to their diploid counterparts. We conducted outdoor field trials at our campus in 2020 with several different diploid, triploid, and tetraploid crossing combinations. Flower production increased between 30% to 100% variety dependent, with the largest leap seen in day-neutral varieties. The yield differences are statistically significant, but the visual difference, honestly, is just in plant size. Larger leaves, larger flowers, thicker stems, denser trichome coverage tells you all you need to know about triploid vigor. There's a slight increase in total cannabinoid content in flowers and a marked increase in trichome density on sugar leaves near flowers. Additionally, olfactory compounds were noticeably more abundant in triploids and tetraploids compared to diploids, but we didn't test for total terpene content in this particular study because terpenes tell less than half the story, and most of the major aroma components of our varieties are not terpenes at all. There's some really exciting work underway on the aroma compounds of cannabis that I promise will upend everything we thought we knew about the topic and give breeders some lofty goals to target in the future. That particular area of research is really exciting. So we have field trials, as Heather mentioned, across the US taking place this summer with a number of different universities, including the University of Vermont, to independently verify these seedless characteristics and to test agronomic responses between different ploidies in diverse microclimates. But the early results from our internal trials in Oregon last summer suggest that triploids are much more resilient to any challenge they face relative to diploid counterparts. There's not much of a surprise that our findings are similar to what have been reported in the literature for polyploid cannabis. Over the past year or so, there's been significant commercial and academic interest in them, from Iran to Canada to the University of Connecticut. Some great publications that have come out, and we're very happy to be contributing to that larger body of literature. An interesting note for farmers, a couple things that we'd like to share. Anybody growing triploids for the first time should know. First, because they produce more biomass than diploids, it's really important to provide them with more nitrogen than you would under normal circumstances. Every farm is obviously unique and requires a soil evaluation, but our general advice is to aim for about 120 pounds of usable nitrogen per acre to attain robust growth. This is slightly higher than the 100 pounds recommended in the most recent UVM study, but it's just because this particular version of cannabis is absolutely mind-blowing for its rapid growth when conditions are right. But you really need to give them what they need and just get out of their way. The second point here is that what you're going to notice is that triploids initially lag behind diploids by about a week or so when flower initiation begins in July. They quickly make up for it with more rapid flower formation and maturation and end up finishing at about the same time. Compliance testing can occur simultaneously with mixed diploid and triploid varieties if they're from our company. For CBD dominant varieties, we recommend that testing occur 28 days before harvest or 4 weeks after flower initiation, when emerging floral clusters are about the size of your thumbnail. The latest reasonable testing date is about 35 days into flower. Any later and you risk failure under the proposed USDA sampling guidelines. Please also remember that the flowering process begins about 2 weeks before any flower clusters are visible. Those first 2 weeks represent the plant's final vegetative push to establish a great flower development platform. The testing warning does not apply to our Triploid CBG or our new Triploid CBD-rich varieties, which can be grown full term and still maintain compliance for the 0.3% total THC. The third piece of advice to be aware of and prepared for are massive stocks on these triploids. You may have to alter your harvesting strategy depending on your equipment. Large farmers need to drive slower with their combines, and smaller farms should buy extra sawzall blades. These are prehistoric stocks. Please also keep in mind that most of a plant's weight at harvest is stored as water in that stock. So drying is going to take longer and most likely take a little bit more space than the equivalent diploid acreage, just because one, you're going to have more biomass, and two, there's a lot of water in that stock. So removing the stem and stock from floral material at harvest is a great way to maximize your drying efficiency if you're able to do that, and maintain the traditional levels of throughput you may be accustomed to. The last bit of advice, and it's an ingrained reaction for experienced cannabis farmers, it's going to be really hard to overcome, but stop worrying about hermaphrodites. These are triploids. They don't waste energy worrying about pollen, and neither should you. The bookend to our polyploid development work is our continued support of several funded research programs in collaboration with Oregon State University, which includes the labs of Kelly Vining, Pat Hayes, and Ryan Contreras. We have research underway there, looking at ploidy induction optimization protocols in cannabinoid-free germplasm, assessing chromosomal stability in induced polyploids over time, genome reduction techniques, tissue culture media assessments of diverse genotypes, RNA sequencing to identify sources of THCA variation in our CBG varieties, and the construction of the first cannabis pangenome derived from our massive collection of whole genome data. The pangenome work is really excited as it's going to be a constantly evolving tool for future generations of breeders to work with and benefit from. This work currently supports 3 master's students and 2 postdocs. In addition to our work with OSU and our field trials this year, we're also participating in a hemp research consortium with Cornell, North Carolina State, and the University of Kentucky. We're really excited to continue working with these institutions to push the boundaries forward in cannabis through good science and endless curiosity. Some important questions for the future will address the best ploidy level for particular applications. We know that cannabinoid production should occur with an odd number of chromosomes to prevent seeding, but we still need to identify which level of ploidy provides the best results for farmers, whether that's triploid, pentaploid, heptaploid, or something even higher. The larger seed size and overall productivity of tetraploids suggests that they would be great for grain production, but their lower pollen fertility may outweigh those intrinsic benefits. Triploids are exceptional candidates for the fiber market, but the actual cost of seed production and the marginal economic returns for that particular end use poses a significant impediment to widespread adoption. Honestly, who knows? I mean, the passage of time and additional experiments should offer some clarity, and I just feel incredibly lucky to be living in the midst of literally the golden age of discovery for this particular crop. So we're really excited to offer farmers this evolutionary leap in cannabis breeding. Triploids we believe, are the future for cannabinoid production. They virtually eliminate the risk of a seeded crop, protect yourself and neighbors from errant pollen, substantially outproduce diploids, and help protect unique production environments from the spoilage associated with feral hemp populations. Their emergence as a viable production plant suddenly makes grain, fiber, and cannabinoid farming possible in the same area. Their innate vigor also makes them excellent candidates for emerging applications like carbon sequestration or bioremediation projects. Our company has released 9 triploid varieties for the 2021 production season, which include CBD, CBG, and CBDV chemotypes, as well as photoperiod sensitive and day neutral traits. These are all certified organic by the Oregon TILT and non-GMO by NSF. So as awesome as these 2021 varieties are, we're looking forward to the new flavors, cannabinoids, flowering response traits, and new ploidy levels to come over the next few years. From large operations like Tyler's to boutique farms like Sarah's, we always have something new in mind to help farmers be successful, even in the face of federal policy instability. I'm really proud of our entire team for making this possible. There's obviously a lot that goes into creating quality seeds at every step in the process, from the foundational science to the final seed production to shipping and on-farm advice. And we have one of the best teams in the business. We're always grateful for our farmers as well. They do a great job making us look good every year. And we love sharing the latest and greatest, especially when it translates to even more successful clients. So for more information, please feel free to visit our website or reach out to either myself for technical questions or our farm relations manager, Emery Garcia, for ordering information. And with that, I'd like to thank you all for your patience with the technical difficulties and say that we're really looking forward to being on the ground in Vermont this fall. So any advice on where to spot burning, please contact me directly.
That's funny. You know, he doesn't live very far from our research farm. And always at the farmer's market that my husband goes to on Saturdays. So Bernie's pretty much there every Saturday. That's just a hot tip.
All right, I've got the pro tip. Thank you.
Yeah. Hey, you never know. You may be able to get them to come up to the research farm when you folks are here too. So, yeah. Well, if folks have questions, we have a few minutes. I know we were just out applying fertilizer to where we're going to plant the hemp sets. So I put on 110 pounds and I guess I'm going to go back and put on a little more. But
are you guys,
yeah.
Do you have drip irrigation out there, or is there?
Yeah, we do have drip.
Okay. Now
So
we can always supplement later on.
Yeah. Yeah. That's a good point. We could do that. It's organic fertility, so it takes a little time to break down. So I was trying to get most of it on now. Anything else? I mean, I was thinking plant spacing doesn't matter so much, or is there anything particular there?
That's a really good question, and I saw that in the chat box. You know, plant spacing really comes down to the type of harvest equipment that you have and the method that you're going to use. We always recommend that people really plan their spacing around that really important component. These plants do take up more space physically. You could get away, normally we would recommend 4x6 spacing, 4 feet in row and 6 feet between rows. But these, you could very easily get away with 6x8, depending on when you get them planted. One of the things that we don't like to do is, give people very dictatorial numbers on that, because if someone plants in July, it's going to be a very different recommendation than if they got in the ground at the right time in June.
Do you have a fact sheet of some kind? I'm sure you do on your website. So people that are.
We do. Yeah, it's
Right,
on OregonCBD.com.
Okay.
We've got a bunch of useful information over there.
Yeah, and oh, great. Thanks, Andrea. Any other questions from folks? I know we're getting on past our time, and I know people are really busy. I had a question, actually, Bob Shipman that was on here, Robert. He was asking me about, this is irrelevant maybe, but just curious about your experience with yeast and mold counts. Are there any specific requirements in Oregon for yeast and molds? And if there are, what are they? And do you have any experience with that?
Yeah, there used to be, but it's a strange policy environment here in Oregon for that. That's actually overseen by the Oregon Health Authority, who manages the testing labs and the requirements that go along with compliance testing for both THC and for industrial hemp products destined for human consumption, but only if it's sold in the state of Oregon. So they used to do total counts, and they used to do plating, as the dominant method of being able to identify those. But there was just a lot of noise in the process of that, and OHA moved to eliminate that a couple of years ago. There's definitely still talk about trying to, maybe bring in a qPCR based approach, as some states are doing, but that part is still kind of up in the air.
Okay. And somebody's asking about that rapid growth, and does it lower resistance to pests and diseases?
Not from what we've seen. It's just overall more vigorous. One of the issues that we have is not being able to do field trials in places where there are, specific pathogens or pests that we don't have here in Oregon, which is kind of the idea behind this year's field trials in diverse locations. But what we saw in our field trials is that they're just more vigorous and more resistant to pretty much anything you throw at them.
Well, that's good. We are in one of those environments where we get a lot of moisture generally, and sometimes disease. So we'll put it to the.
Awesome. That's what we need.
Good.
There's some indication in other crops, just to throw it out there, there's some indication in other crops that polyploidy can actually increase disease resistance. So it'll be interesting to see what happens with cannabis in general as we have more and more locations where they're grown out.
Thanks, Andrea. Great. Well, I think we're right at time. And yeah, again, apologize for the technical difficulties. It happens. And yeah, we really appreciate your time. We know that you're all very, very busy at Oregon CBD, and it was really great. The triploid varieties, and hopefully you'll make it to Vermont, or some of you will this fall, and we'll have a field day, and we'll invite Bernie, see if he can make it. So
you're making my day. Thank you so much.
At least we'll have a meme of him here or something. There's a lot of those. So. Well, thank you, everyone. And like I said, we'll video or tape it. It's been recorded, and we'll make it available. I know there are a lot of folks who come today and are out in the field. So yeah, everyone have a great day. Seth and Andrea will be talking to you.
Sounds good. Thank you.
Thanks,
Bye,
Heather.
Andrea. Bye.
Bye. We'll see you soon.
Thank you.